Hello everyone, welcome to BioOcean and this is me Sheethal with you, and today we are here with our new video and the topic is all about ribosomes, which is a cell organelle, and this video is again a part of cell series. If you haven't watched our cell series, please click on the I button. I have attached all the videos in the series. You can access and understand the whole concept there. So today we are focusing about one of the major important cell organelle called ribosomes. And starting up with the introductory part, we can say that the term ribosome was proposed by Richard B. Robert in the year 1958. And this ribosome is made of complexes of RNAs and protein and together called as ribonucleoprotein. And each ribosome has two subunits, a larger subunit and a smaller subunit. The larger one binds to tRNA and amino acids while the smaller one binds to the mRNA. And after reading a mRNA molecule, these two subunits split apart. And then comes about the two different types of ribosomes, that is prokaryotes have their own type of ribosome while eukaryotes too have their own type of ribosome. So let's understand first of all about the prokaryotic ribosome. It has a diameter of 20 nanometer and is composed of 65% of ribosomal RNA and 35% of ribosomal proteins. While in case of eukaryotic ribosomes, the diameter relies in the range of 25 to 30 nanometer and the ratio of ribosomal RNA to protein is close to 1. And the ribosomes translate the messenger RNA or the mRNA and build polypeptide chains like proteins. We can say an example like proteins. And they use amino acids delivered by the tRNA. Active sites of these ribosomes are made of RNA and so they are classified as ribozymes. And here the measurement unit is called as Swedberg unit. And now let us understand the prokaryotic ribosome as well as eukaryotic ribosome in detail. First of all, let's start with the prokaryotic ribosome. It is called as a 7DS ribosome, that is the small subunit is 30S and has 16 sRNA, about 1540 nucleotides bound to 21 proteins, while as in the case of prokaryotes, the larger subunit is 50S and has 15 sRNA, that is about 120 nucleotides, and 23 sRNA, about 2900 nucleotides and 31 proteins. And then comes about the eukaryotic ribosome. Eukaryotic ribosome is 80S ribosome and it too have a small subunit as well as a large subunit. The small subunit is of 40S and has 16 sRNA, about 19,000 nucleotides and 33 proteins. While as the large subunit is 60S and has 5 sRNA, about 120 nucleotides and 28 sRNA, about 4,700 nucleotides, and 5.8 sRNA, about 160 nucleotides, and 49 proteins. Various ribosomes share a similar core structure, and RNA is highly organized into various tertiary motifs. And catalytic activity of this ribosome is by RNA. Protein, which is seen on the surface, is the stability structures is used to stabilize the structure. And now let us understand the functions of ribosome. As we all know, it is called as the protein biosynthesis part. Otherwise, we can call it as the workhouse of protein biosynthesis, that is translating mRNA into protein. And mRNA comprise a series of codons that dictate to ribosome the sequence of amino acids needed to make the protein. And using mRNA as a template, ribosome transfers each codon of mRNA, pairing it with appropriate amino acid provided by tRNA. The molecules of tRNA contain complementary anticodon on one end and appropriate amino acid on the other end. Small subunit typically bound to tRNA contain amino acid methionine, binds to AUG codon on mRNA and recruits the large subunit. If you have to know more about amino acid, you can please click on the I button. I have already made a video on that and you can access it there, okay? And then the ribosome contains 3 binding sites called A, P, and E sites. A bind with amino acyl tRNA, P bind with peptidyl tRNA, and E binds free tRNA before it exits the ribosome. Protein synthesis here begins at the start codon AUG near the 5' end of mRNA, and mRNA binds to the P site of the ribosome first. P site of the ribosome first. And in bacterial cells, several ribosomes are working parallel on a single mRNA, and that is called as polyribosome or polysome. More than one ribosome may move along a single mRNA chain at one time, each reading its sequence and producing a corresponding protein molecule. Once protein is produced, it can then fold up to produce specific functional 3D structure, and it is determined by the pattern of charges in its sequence. These are all the functions of ribosomes, and we can say that they all are interconnected. And before the functions, we understood about what like a ribosome is, how do the eukaryotic as well as the prokaryotic ribosome differ from each other, and what all are the constituents of each ribosome, and like that. I hope you get an overall idea of what a ribosome is, and I hope this video was very helpful for you. If you find this video more effective, please do like, share and subscribe. And if you have any kind of doubts or suggestions, please let me know through my comment section. And please do share this video with everyone who is interested to learn biology. Please do share my channel, and thanks for watching. We will be coming with the next exciting video. Thanks for watching. God bless. That's all for today. Thank you.